In this lesson, we are going to explore how different biological molecules found in the food we eat, such as carbohydrates, proteins, and lipids, are broken down during digestion by enzymes into smaller, soluble molecules that our bodies can absorb. This breakdown is essential because the nutrients we consume in their natural form, like starch, protein, and fat, are often very large molecules, far too large to be absorbed directly into our bloodstream through the villi in our small intestine. Instead, they must be broken down into smaller, simpler units, and this is where enzymes come in. Enzymes are specialized proteins that act as biological catalysts, which means they can significantly speed up the rate of chemical reactions without being used up themselves. Each enzyme is highly specific and targets a particular type of molecule, ensuring efficient digestion. Let's now look at which smaller molecules each type of nutrient is broken into and which enzymes are responsible. Starting with carbohydrates, the majority of these in our diet come from starchy foods like bread, rice, pasta, and potatoes. Starch itself is a polymer made up of many glucose molecules joined together in long chains, and each individual glucose molecule is known as a monomer. In order to digest starch, the body uses an enzyme called amylase, which breaks the starch down into a simpler sugar called maltose. Maltose is made of just two glucose units bonded together. To complete the process, another enzyme called maltase is used to split each maltose molecule into two glucose molecules. These glucose units are now small enough to be absorbed into the blood and used for energy in cellular respiration. An interesting and useful pattern to notice is that many enzymes have names ending in the letters ASE, such as amylase and maltase, which makes them easier to identify and remember. Now, let's move on to proteins. Proteins are found in foods such as meat, eggs, legumes, and nuts, and they play crucial roles in growth and repair in the body. Proteins are made up of chains of amino acids, which are the building blocks of all proteins. To digest these proteins, the body uses a group of enzymes called proteases. These enzymes break the long protein chains down into individual amino acids, which are then absorbed and used to build new proteins needed by the body. There are several types of protease enzymes, including pepsin, which is active in the stomach, and trypsin, which works in the small intestine. While there are many different amino acids, you're not expected to memorize each one individually for most biology courses, but just understand that they are all small enough to be absorbed once the protein has been digested. Finally, we come to lipids, which is a term that includes both fats and oils. These are found in foods like butter, oily fish, seeds, avocados, and cooking oil such as olive oil. Lipids are digested by enzymes called lipases, which break each fat molecule down into glycerol and fatty acids. Both of these smaller molecules are soluble and can be absorbed through the walls of the small intestine. In addition to lipase, the digestion of lipids is helped by a substance called bile. Bile is not an enzyme, but plays a critical supporting role. It is produced by the liver and stored in the gallbladder, and it helps by emulsifying fats, which means breaking large droplets of fat into many smaller droplets. This increases the surface area available for the lipase enzymes to work on, making the breakdown of lipids more efficient and quicker. Before we wrap up, it is also essential to understand where these digestive enzymes are produced within the body. All three major types of digestive enzymes, including amylase, protease, and lipase, are produced by both the pancreas and the lining of the small intestine. In addition to those, amylase is also produced by the salivary glands located in the mouth, which is why digestion of starch actually begins as soon as we start chewing. Proteases are also produced by the stomach, which secretes gastric juices containing the enzyme pepsin to start breaking down proteins in the acidic environment. Understanding both the function of these enzymes and their origin points in the body helps to form a complete picture of how digestion allows our bodies to transform complex foods into useful, absorbable nutrients.